Okay, hi everyone, how are you? Last week we have looked at, in this chapter, we have looked at a classical viewpoint, what they think about the concept of gene. So, according to that classical concept of a gene, which was first formulated by the renowned Gregor Mendel, the Catholic priest in 1800s something. What we can interpret is nowadays what we can interpret his view about the gene is the gene will create all different traits. Right. So gene is the one ultimately responsible for different traits. Right. But no one knew about what exactly the, so to speak, the molecular nature of the gene. Okay, there is a gene and something that is something very mysterious. That thing appears to determine all those the different traits in the offspring, so that this gene thing can be passed on to the next generation after generation after generation, and then it works like a mathematically accurate those pattern of inheritance. So anyway, that worked. So that's the whole thing about the beginning of the new branch of this biological study we call the genetics, right? So he founded the new branch of a study in life science, which we call the genetics. However, in this modern genetics, which incorporated the element of molecules now, the Watson and the Crick, the contribution was actually what exactly was the gene itself. And then it's the DNA kind of molecules. Oops, that's it. So then now we have an opportunity to have a more detailed, conceptually speaking, more detailed idea about what genes are like and what they do actually in terms of the function. So if we now try to use this modern viewpoint to define gene. So by the way, so like all those traditional genetics as I introduced in last week as a like transmission genetics, has been changed into new terminology of after incorporating those chemically the molecular aspect of so DNA is nothing but all these nucleotides, polymer of nucleotides, right? So that's how we know about the nature of the gene. Wow, gene is simply nothing but a whole the assembly of little tiny bits of different chemical molecules we recognize as nucleotides. Is that it? Then how do they do? How do they work to control? Still nothing changes. Nothing changes that those gene things, those gene control the traits. If the gene changes, then trait changes. Okay? What do you mean by gene changes? Now we understood. Before, we didn't understand how gene changes, but something changed. Some changes occurred in the gene, and then as a consequence, the trait, corresponding trait also has been changed. Wow. So there's a tight link. So if you want to change your trait, whatever if you want to improve or something change into what as your desired pattern, then there is no way other than changing your gene. This tight correlation. Gene will dictate. Okay. That still stays true in that even if we found out that gene is nothing but the DNAs, the whole thing, the DNA sequences, so to speak, the nucleotide, some specific orders of nucleotides. So we knew that what do you mean by the gene changes? That mutation now, we use the new terminology of what do you mean mutation? Some DNA sequence change. Original order of this nucleotide somehow altered, okay? Something is missing or something was added or this order was simply just changed. Then altogether we call it a mutation, and the consequence of the mutation is changes in traits. So nothing changes, but we knew better about the nature of the change in genes. So in molecular genetics, there are two definitions in molecular genetics, definition for genes. One narrow definition is I simply will read segment of DNA that carries information for protein synthesis. Okay, so now the bottom line is this: protein. That's the focus of this lecture, protein. So now we kind of want to familiarize their tight natural link between DNA, the gene, DNA as a gene, and the protein. As I went over last week, if the gene as a DNA sequence were some kind of recipe for making pizza, then protein is actual pizza so that we can taste. And then actually those pizza can help us by providing the nutrition that we need. We cannot eat the recipe for making pizza, although it is really great and valuable, but simply by just eating up the recipe doesn't do anything for us. We have to eat the pizza. So that's the analogy for the relationship between the DNA, the gene, and the protein. The value, the reason why we so value the DNA, I mean the gene in the DNA so much is each gene will generate their particular respective version of proteins, and proteins are the one made possible for everything in our everyday living activities, right? So, so it's fair. It's fair to define as it is like, oh, what's gene? Then now we can, oh, there is a segment of, what do you mean segment of DNA? In other words, like as we looked at it last week, again, along with our genome, entire DNAs in our cell, not every area of our entire DNA serve as gene, but only some fraction, some certain specific area of our whole entire DNA can function as a gene. So that's why we have to say like segment, not every DNA, but only some part of the DNA. What part? In what way? Functionality will distinguish the difference between gene and intergenic region. The gene can carry the information for protein synthesis. Okay? The intergenic region, the rest or garbage area, actually some in the past, some time ago, the scientists actually thought those intergenic region as simply the garbage area, useless, because they don't contribute anything in making this protein synthesis. So they don't function as any gene. So what's the use for? Yeah, that's the DNA, fine. We'll accept that. We carry them, but simply those are kind of a trash garbage area. But it turned out that no, it is not. Even the intergenic regions are also very valuable and important for the overall activity and the viability of our genes. But anyway, so that's how we can say about it. But that's what did I say? That's an only narrow definition. What's then the broad definition? So this is the cartoon, and I took it from one of the old biology textbooks. So the gene, and the relationship between gene and the protein. Like genes is the information that signify, which we call code. Genes are the one that some particular order of this nucleotide. We abbreviate a nucleotide each. We have only 4 nucleotides, right? As if, like in our vocabulary of genetics, we are very poorly equipped in that we use the language which consists of only 4 alphabets. Wow. Even English has a 26 alphabet. Jesus. And this, our genetic code, have only 4 alphabet. How can we use this only 4 freaking letters and do something meaningful? But it turns out that, wow, it's so actually more than necessary, really valuable, meaningful job for us. But anyway, this 4 letter, we are kind of assembling each different vocabulary word to create any meaningful sentence. So each amino acid, somehow, that's what we are going to learn this chapter. Somehow, with the combination, different assembly and combination of the 4 alphabet, and somehow generate 20 different vocabulary. Okay, each word, each amino acid. We already knew that proteins are basically the polymer of different amino acid. How many different kinds? 20 different kinds of amino acid, right? So each amino acid we can view as each different word, vocabulary themselves. So we speak language using 20 different words, very limited language expressibility, but still, anyway. So any one particular version of a type of protein, we can think as a kind of a sentence, yeah, made up of some different combination of words. That's the relationship probably we can think as a. So anyway, it all begins with this DNA providing the alphabet. And somehow okay, I will tell you what. With 4 different alphabet, in order to signify each different word, which is amino acid, we use 3 nucleotide unit as a one kind of a such alphabet unit to designate each different amino acid. So that's how we can, with using this, somehow we can manage all different living organisms. Same thing, same principle, same mechanism. Somehow can manage it to generate 20 different amino acids. In between lies the RNA. RNA is simply, we can view as a kind of messenger boy, bellboy. As I said last week again, this information, the recipe is really precious because this is the only single copy. If we lose or if we damage this, we are doomed. So we cannot let anybody simply use freely this our original copy. So if someone wants to have an access for this original holy the information, we better let them use some kind of Xerox copy. Hey, why don't you make some copy so that I can still stay safe? That's the nature of the RNA. RNA simply copy the original information and then bring to the site of actual cook place. Hey, let's make pizza. So what's the recipe? So and then we follow the instruction. Then voila, the pizza is there. Okay, so in that regard, so each gene we can view as to define what's gene. Then any segment of information that can generate a sentence, right? Protein, right? But then, was there to be any broad definition? Isn't it enough? Well, we understood now. Okay, and why do we have to once again worry about any unnecessary broad information? Because unfortunately, some RNAs, I mean, some information, some DNA information does not directly generate any kind of a protein. Oops. So these are, what do you do? What's your role? Well, I make RNA. Fine. And then does your RNA now eventually generate protein? Oh, nope. And what do you have to be there? But there are so many, many such RNAs do also exist in our cell. So how do we handle this? Those guys do not contribute anything, at least apparently, doesn't seem to be contributing anything in actual the protein generation. Protein is our own main interest because protein is the food. Protein is magician. And if anybody is nothing, doing nothing regarding the protein production, why do we have to care about them? But there are so many such DNA information. Directly, they are not actually related with in giving any protein synthesis information, but still they generate RNA. Because this is DNA as a gene. According to the broad definition, DNA, then generate the RNA. Because the relationship between DNA and RNA is what did I say about this RNA? RNA is a copy of the DNA. But in reality, practically speaking, how do you make copy? If there is a DNA, you want to make a copy? How? It's actually complementary. Do you still remember this relationship, this complementary base pairing? So like if DNA is a double-stranded, mind you. So let's say if the segment of DNA is AG, CC, but DNA doesn't exist like a single strand like this. DNA, TC, GG, TC. This is DNA. But RNA is a single strand. So RNA will make copy. Which one? And how? Always, this nucleotide, whether it is an RNA or DNA, they are basically chemically same material, which is nucleotide. The beauty of nucleotide is they can form the complementary base pair with specific partners. So, how do you make RNA? Simple. Look, so you choose, hey, I want to use this information as a gene, right? The top one. Then you try to copy this one, but in doing so, you are not directly generating a copy, but you have to first generate complementary thing, right? So if you want to pick this information among these two strands, then you generate complementary of this top strand, which is as an RNA, what? You read. Can you read? RNA, A. What's the complementary to A? T, but remember, it's an RNA. U, right, U. And C, U, C. And GG, U, and C. So this is the RNA generated if this was the information that served as a gene. Then this gene will first generate RNA going like this sequence. And usually this RNA will go further, be processed to generate protein, and because each three, whatever, this information will be matched to specific amino acid 1, amino acid, I abbreviated amino acids AA, amino acid, and like this. So that's how you eventually generate protein, because amino acid, you can link each amino acid to assemble a protein. However, a lot of, so to speak, this DNA or gene, according to the broad concept, broad definition, they simply stop here. They simply stop here and I'm done. I don't have any business further. Huh? I thought you were going to go through this next step to give us a protein and then you stop there. What are you? I am still gene. Oh yeah? So that's the kind of a dilemma or conflict. So that according to the narrow definition of the gene, oh, you guys are not gene because you are not producing any protein. However, there are so many such that ends only as an RNA. So then do we call them as a what? The garbage? Oh. So then we have to make some kind of a compromise. Okay, so how about defining gene as a rather broadly? So broad concept of the gene is any segment of the DNA that can now make RNA. But here, another important thing is this. Hey. Can make RNA. Every one of you can become an ace in this classroom. But not everyone will be an ace in the classroom. That's the difference. Yeah, everyone has a potential. But just because you have a potential doesn't mean that you will or you will have to. Same thing. Segment of the DNA they can make does not mean that they will always make RNA. They have an ability. They have an ability to generate RNA, but simply it's up to them. Well, I don't want to. I don't feel like. Then fine. Yeah, take some rest. But still, I'm gene. So that's the definition. Oh my gosh, my mom. 여보세요. 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 Whatever. Okay, so here we finally came to this point that what's gene, then even we can say that any area, any defined area that potentially can generate RNA. Now it's a good time to consider, okay, how many different RNAs are there in our cell? And why do we have some different types of RNAs? Isn't RNA always the same RNA? No, unfortunately, because there are several different kinds of RNAs out there, each functioning differently. So one major RNA that we rather actually want to focus more because it is directly related with the protein synthesis. It's called mRNA. M stands for messenger RNA. Okay. So it's another name, another way of recognizing this mRNA is they are coding RNA. What do you mean coding? Because the information written in this messenger RNA will always have some kind of meaning in defining some specific amino acid. So this mRNA is the one that actually will generate pizza for us. So those are the ones that fits into even the narrow definition of the gene. If any gene generate messenger RNA, then definitely they are gene, both in narrow and broad definition categories, okay? Because it will generate protein. What else? So that is messenger RNA. Another very important RNA is a ribosomal RNA. We have already looked at the ribosome as some mixture of some kind of RNA and the proteins. And the importance of ribosomes also we have looked at it again, because without ribosome we cannot do anything, because it is the machinery. Ribosomes are the machinery that synthesize, produce protein. Okay? This is the only place. Ribosome is the only place that we can synthesize protein there. But in order to assemble ribosome, we need definitely ribosomal RNA, some as a very critical ingredient and component of the whole mixture of this thing we call ribosome. Okay, but since ribosomal RNA does not signify any specific amino acid information, unlike messenger RNA, we call that as a non-coding RNA. Some may think that, hey, non-coding RNAs is just unimportant because you do not do anything in making protein synthesis. So why do I have to care about you? You are just a loser. I will only care for this, the coding RNA, which is messenger RNA. If that is your what you think, then think again, because without ribosomal RNA, what can we do? Nothing. We cannot do anything because we cannot do the actual protein synthesis. So just because they are non-coding RNA, we cannot ignore them or despise them is the thing that we have to accept. Okay, so ribosomal RNA is an example, such one example of non-coding RNA. Fine. What else? tRNA. T stands for transfer RNA. Once again, it is a non-coding RNA, right? And this is a delivery guide. To synthesize this amino acid protein from amino acid, somebody has to bring each different amino acid to the site of a protein synthesis, which is where? The ribosome, right? Amino acids are scattered all over the place in our cytoplasm. But ribosomes are specifically in some place. Like, for example, some ribosomes are on the surface of the endoplasmic reticulum. Some ribosomes are some specific area of the cytosol. Yeah, not everywhere. So to each of those ribosomes, each different, 20 different amino acids must be provided so that all those raw material, enough raw materials are at least provided so ribosome do not have to worry about getting the supply of any raw material. That job is carried out by this tRNA. Each different transfer RNA will pick up the amino acid. So at least if there are 20 different amino acids are there in our cell that participate in the protein synthesis, how many different types of transfer RNA do we have to have? 20 at least, right? Each different tRNA will take care of each their respective partner amino acid. Okay, those three RNAs are probably some of you who took the minor, like a mild biologicals and your high school or somewhere else, then probably those three types of RNAs are the one you probably most probably at least most definitely heard of. But these three different type of RNAs are the one that actually newly emerged RNA. So 20 or 30 years ago, people didn't know about actually that's there. But now in these days, oh, there are some kind of these RNAs too. One such RNA is miRNA, which is similar to mRNA, but this is not a typo. Mi stands for microRNA. Yeah. All RNAs are micro, but these RNA are more micro, so that we have to call them as a microRNA. It is non-coding RNA too. But the importance of this non-coding, well, we already knew that the importance of ribosome RNA and tRNA, even though they are non-coding. So yeah, there's no point of like despising any RNA just because they are non-coding. But anyway, this microRNA is non-coding, which means they're not directly involved in any protein coding, protein synthesis. However, it's very important in that it's kind of a controller of gene expression. What's gene expression? I every day casually, because I'm a DNA guy, every day I casually speak this vocabulary as a gene expression, gene expression. But to you, what's gene expression? What do you mean, gene express? Hey, I'm here. That's gene expression. I have some feeling. That's the expression. Gene expression means making RNA is a gene expression. By the way, the terminology. The process of making RNA, the gene expression, we call it a transcription. Actually, this transcription is kind of like a reminiscent of copying things, copying transcript. Because when you're in college and you want to have your proof of your academic record, and then you call to the registrar's office, and then, hey, can I have my transcript? That means your 성적표, right? Transcript. The copy of your whole academic record is called the transcript. So transcription means copying things. Your original record is kept in where somewhere really very secure place, like a registrar's administrative office or anything. But you only deal with the copy. That's why it's called transcript. And because it is making RNA, this business is simply copying the DNA information. That's why it is called transcription. So anyway, gene expression means transcription, right? Which means making RNA. And so this business is very important in order to generate enough and needed critical kinds of protein for us. And who controls this? These guys control this. Wow, who are you? Yeah, this microRNA do that. And these things have been discovered very recently. Oh, why you are here? I didn't know. Hello. She is the one, actually, the world-renowned expert of this microRNA business. So she's a professor in the Seoul National University, and in this molecular biology, especially in this RNA control, microRNA world, no one, everybody knows about her because she is one of the top world-renowned scientists in this area. Okay, and another RNA, snRNA, is called the small nuclear RNA, which is also non-coding RNA, removable introns. My kind of enthusiasm is toned down a little bit because it's a little more complicated things and important. I'm not saying it is not important as others, but I would rather actually stay away from the main as a main discussion of simply is there another kind of such non-coding RNA and also another kind of non-coding RNA, snow RNA, whatever. Okay. So anyway, bottom line is there are several, altogether 6 kinds of RNA that we have, but there are some more. Even I did not even bother to include this the list of the different kinds of RNA from the classical molecular biology that we knew back then there were only 3 different kinds, but now it has been extended into 6 major different kinds of RNA, which even can be easily expanded to something more. So the bottom line is there are many different kinds of RNAs are in our cells, and one other thing is only one of them is coding RNA, which is messenger RNA, right? Everybody else is non-coding RNA. But just because non-coding RNA, we don't have to worry about them? No, because they are also equally important in providing us with many different functionality. Okay? Now, uh, so we do have whole DNA. Like in terms of by counting the nucleotide wide, we have more than 3 billion pairs of nucleotide, more than, which means in terms of the nucleotide numbers, it's more than 6 billion nucleotides, right? Along with such a long thread-like thing of this DNA, only very small fraction of the areas are actually engaged in this RNA making activity, whether it is a coding or a non-coding. Making RNA activity, we call what? Transcription, right? So only very small areas are actually being transcribed. Then what's the rest of those areas? What do you do? So those area, many area, majority of area that are not actually doing anything in terms of this gene expression. Okay, they have some kind of a common characteristic denominator as they form so-called heterochromatin. So here I'm going to introduce, although it is something a little bit difficult concept, but in these days, this concept, the all information about these genes, more and more spill into general public. So probably it's not really such a too bad idea about trying to distinguish the difference between heterochromatin and the counterpart of this heterochromatin is we call the euchromatin. So that's what we are. That's what I'm going to introduce to you. So anyway, this area is called heterochromatin, and if this thing, this whole cartoon represent one diagram of a particular chromosome, uh, like generally, roughly speaking, the heterochromatic area, the most renowned heterochromatic area is this central region, like your belly button area, and the two edge, top and bottom regions are very famous heterochromatin area. And those heterochromatin areas are, uh, they do not produce any gene activity. They do not produce any transcription. Another area, so the rest of the area, okay, the central, this area and this area, we call as a euchromatin. So except for this, so this and this probably, these are the euchromatin area that are we can find more frequently find the activity of the genes. They are more frequently transcribed. Okay, so we are going to get into that. This is the difference between euchromatin and heterochromatin more today. But first off, this heterochromatin area, like this are three major, very famous heterochromatin area. Okay. The DNA sequence are more highly variable in each different individual. Hey, our DNA, human DNA, is 98 point about 9% identical to that of chimpanzees. 98.9%. 98.9% is almost 100%. So that's the kind of the comparison. If we read our DNA sequence of human, representative human, and if we also read the representative of chimpanzee's DNA sequence, it's like identity is something like more than 98%. Wow. Almost identical. Still, everyone knows that no one have any mistake for chimpanzee, for human, or anything. They are too different. And those differences are like DNA sequence differences are responsible for these things. So in other words, even with very such a small difference, are enough to generate such a distinct differences among different organisms. But speaking into the within species, among different humans, every different humans, we all know that we are all different. And why are we so different? Because our DNA sequence is not identical, right? We are not identical twin. If we were all identical twin, then we will look alike, and DNA sequence will be basically identical. So even among different individuals, we do have enough variation, DNA sequence-wise. Probably it will be more than 99%, the identity-wise, the similarity among different individuals in terms of DNA sequences, more than 99%. However, still, with that only 1% or less than 1% difference is just more than enough to differentiate all these different individuals. But speaking of this variability, okay, this variability in each different chromosome, the whole entire area of chromosomes are not equally like the same way distributed equally, so that in terms of like on average, if we have a difference in 1% difference, that 1% differences are like scattered, evenly distributed along with all different 23 different chromosomes. No, it is not. There are some areas, each different individual will carry more individual differences than the other area. And this so-called heterochromatin area is that we can characterize this heterochromatin area as more variable. What do I mean more variable? If we compare DNA sequence among different individuals, right, then the difference come more frequently in this area than the other euchromatin area. Why? Why? Why do you think? This is a very important question. Why do you think the heterochromatin area has more individual DNA sequence variation than the other so-called euchromatin area? Anybody? Incentive, 2 points. Should I raise in this area of inflation? Should I raise this a little bit, like a 3 point? No, that'll be too much. 2 point. 2 point. Anyone hit? Well, gladly to give you a 2 point. Why do you think this heterochromatin area has more sequence variation in different individuals than the euchromatin area? To answer that, probably you have to first consider what's the difference between heterochromatin and euchromatin, right? What's the difference? At least according to the information that was provided to you until now. Trying to say something. Euchromatids are like having more genes, right? Heterochromatin, they don't have any genes usually. That's why in heterochromatin area, you seldom find any area that is engaged in RNA synthesis, RNA making. That means in heterochromatin area, you don't find any genes, hardly, right? Usually the genes, or most of the genes are accumulated, okay, on only the euchromatin area. That's how it is. Okay, having said that, so the difference between heterochromatin and euchromatids are gene poor versus gene rich, probably, right? Heterochromatin area, you don't really hardly ever see genes. Euchromatin area, you get to see a lot of genes. That's the difference. With that, then why do you think this heterochromatin area has such a more degree of freedom, more freedom of being different? DNA sequence variation. DNA sequence variation comes from what? What's the source of DNA sequence variation? Let me ask you again. What is the origin? What's the source for DNA sequence variation in each different individual? Mutation, right? Mutation, right? All this DNA sequence variation, like we can assume that although there are some other reasons, but mostly the reason why our DNA sequence became different, even though we were all originated from the common ancestor, but nowadays the reason why we are so different in terms of our DNA sequence is because of the mutation. But then, didn't I say some time ago that mutation occur randomly? Right? Mutation will occur randomly. Then if the DNA sequence variation all come from mutation, why there are some inequality? Some specific area, which is a heterochromatin, has a high rate of mutation, and as a result, they present more sequence variation in individual, and euchromatin area has such a low mutation rate, so generally DNA sequence variation is very low. Why? Why is that? Answer is evolution and natural selection is the answer. Still, mutation will occur randomly. But let's consider the consequence of the mutation. Let's say a mutation hit the one such area where your gene, like the hemoglobin gene was there and mutation occurred. what would be the consequence? You die. Because if you have a mutation on the hemoglobin gene, so that consequence, as a consequence, usually the consequence would be the hemoglobin gene will lose the function. Yeah, you are lucky that you are diploid. So even if you lose the function, that the other copy, which is still hopefully functioning normal, can cover up. So you can still, but what if your children's, there is a chance that your children will hit these consequences because of this combination. Now, this mutation become homozygous, so then you die. Because this hemoglobin function is so critical. Most of the genes function will be like that. If you have a mutation, like the end result of a mutation is usually undesirable. DNA sequence change. If you change something, then the chances are it is more likely than not that you will lose the function. Yeah, sometimes you hit the lottery. So yeah, as a result of this mutation, you become better. Yeah, sometimes, but not everybody. Those lucky break will not come to everyone. So generally speaking, if you have a mutation on your area of the gene-rich area, the consequence will be, oh, you're not good. So you will have a much less chance of for your reproductive success. So those who had the mutation, unfortunately, on the gene area will be eventually will be phased out, okay, in this business of a competition under the this natural selection. But yeah, mutation will still occur randomly, but those mutations occurred on those heterochromatin area. There are no genes. So you're free to accept everything. Because so what? Yeah, DNA sequence changes on this heterochromatin area. So what? Because nothing will happen. So you can just keep going on. And as a consequence, over several billion years later, now every different individual will carry much more huge variation on accumulated on this heterochromatin area than the euchromatin area. Those who had accumulated the mutation on the euchromatin, the gene-rich area, are no longer with us. We are all survivors. And as a survivor, we better have preserved the integrity of major functional genes. So we better not have any deleterious mutation on our gene area. But those area which we don't carry any gene is a totally different scenario. Yeah, we can allow as many mutation pile up on those areas. That's why, as a consequence, that's why in these days, if we examine each different individual, so actually, if we compare the DNA sequence of our hemoglobin gene in every different individual, the identity of DNA sequences are strikingly almost identical, usually. Yeah, even with some rare occasions of mutations, like including sickle cell anemia and blah, blah, but overall, and all other important gene DNA sequences are like that. Otherwise, yeah, we wouldn't have been here. But those area, heterochromatin area, which we do not carry any genes, yeah, we can have many variation of DNA sequence as we will like, and that's we are. So what's the point? So what? Because nowadays this DNA sequence, DNA fingerprinting kind of this DNA profiling, using DNA sequence comparison, and then we identify, okay, you are my son, oh no, you are not my son, kind of these things. DNA profiling is actually, we are taking advantage of this area, of this heterochromatin area. Yeah. I mean, if we compare this some gene-rich area of DNA sequence, how can we distinguish anybody? Yeah, impossible. But if we just like fingerprinting, so that's why this DNA profiling is in many occasions it's also called DNA fingerprinting. Just like our unique fingerprinting, this area's DNA sequence is a kind of a hallmark for each different individual's identity, thanks to this free sequence variation of this area. Okay, so uh, forget about this different terminology. This satellite DNA and VNTRs are some specific DNA sequence motif and area that we particularly utilize this particular type unit of DNA sequence in this DNA profiling, but that's a rather more professional way of. So when newspaper talks about newspaper, oh, I didn't know newspaper can talk, but anyway, when newspaper talk about satellite and VNTL, then okay, those are kind of, but you don't really have to be so serious about knowing what are really there unless you are particularly individually interested in it. Oh, shoot. So, and this heterochromatin area, we carry a lot of this heterochromatin area. So these heterochromatin areas are not essential actually because we do not have any genes there. It's just simply there. Well, thanks to them, we can utilize them as like a identification marker, like a DNA fingerprinting. But other than that, but if we really think about it, why are there still, like in terms of this evolutionary race, if we just, it's kind of ridiculous sometimes that we still keep this garbage area. Like everybody wants to become more efficient. If we carry this garbage area, we probably it's like counterproductive. So someone, yeah, legitimately will ask that why do we still keep this non-essential heterochromatin area? The answer is no one knows, but it's there. Someone says actually it's also rightfully so that it is kind of a scar of the all this battle between human and these viruses. Like over the long history of like human evolution and also animal evolution and plant evolution too, or the same. Uh, Virus can come in and out. All this virus DNA sequence can snake into some specific area of our gene, and then they just pop out, and they keep repeating these things, and then ruined everything. So those areas became such a garbage, jumbled, meaningless area, but we still we cannot do anything about it. We simply just cut it off and then dump it. But there is no such uh, quick, efficient means of removing it. So probably we just still carry it. We don't know. So this particular technology of DNA fingerprinting, although I would like to discuss these details of these techniques, but no, I rather not, because a lot of when I did that in the past semesters, when I went over this specific individual techniques, it's not really warmly welcomed by the students. They were, oh, I don't understand. I don't understand. Okay, fine. Okay. So it's there. DNA fingerprinting is there, and it's simply comparing our this highly variable area. Let's just leave it that way. So uh, there are really interesting aspect of, and also technically speaking, if we understood, then you can understood better about some basic principle of molecular biology. So it's a shame, but to go over this, it takes a lot of the technical issues. So I rather just refrain myself from overcommitting. So uh, uh, I should I should skip this. Oh my. Okay, well done. So now, uh, so let's talk more about this heterochromatin. So there are two different areas now. Let's assume that at least now we agree on that along our chromosome structure. Yeah, at least we can distinguish by now that the difference between chromosome and chromatin, right? Chromosome is the highly condensed form that we only usually get to see them just right before the cell division. And for the rest of the whole area, the period, chromosome exists as a chromatin, right? But for those chromatin, there are two different kinds of chromatin is now the new information that I just spilled today. One is called heterochromatin. The other one is a euchromatin. At least one difference between heterochromatin and euchromatin is in terms of what you carry is a heterochromatin area carry almost no genes, whereas euchromatin area carries a lot of genes. That's at least something that we know up until now. Now let's look at the difference between hetero and euchromatin in terms of their actual structure, not what you have, but now how you made up all, the structurally speaking. In doing so, we are going to take a look at this new aspect of this chromosome structure as a dynamic nature of chromosome in that um, first chromosome is like so it's no secret by now. Chromosome is half the protein and half the DNA, right? So this chromosome, entire chromosome, is like bound with this particular protein called the histone protein. And their organization unit is called the nucleosome. So there's once again to remind you the difference between chromosome and chromatin. And actually, if you zoom in this chromosome structure, each chromosome, like wrapped up, the protein, the histone proteins, form a core of a ball-like core, and those ball-like core unit we call as a nucleosome, around which DNA are wrapped. Okay, and in doing so, this nucleosome have a different distance, like accordions. It can be expanded and then shrunk, and like that in three-dimensional way, not two-dimensional way. And that's how you achieve such a great degree of condensation and expansion, loose and condensed. Okay. So that's the difference between this chromosome and chromatin. And that's the difference actually, physically speaking, that's the difference between heterochromatin and euchromatin. So even at the stage, in the stage of a chromatin, you can have a varying degree of this condensation. Heterochromatin area, you have a more tightly squeezed this. So the distance between each individual nucleosome unit is shorter. Whereas in the euchromatin area, they are like more distantly spaced, and that's how they achieved more relaxed, uncondensed version. So this, to give you only bottom line, heterochromatin, up until now, we just view this heterochromatin as those areas do not carry as many genes, right? And they are highly also condensed region. So once again, permanent intrinsically, let's say, although this kind of structure can be only seen in just right before the cell division, but if we view as a chromosome in this way, this central area, like a belly button area, like central area, and the top area, these two, these are the intrinsic or permanent hetero area. So these are always, just like a permafrost, always frozen area, they are intrinsically, permanently, almost permanently heterochromatin area, highly condensed. So the rest of the area we call as a euchromatin. Rather softer area. In terms, what did I say about the difference between heterochromatin and euchromatin? Even as a chromatin stage, these heterochromatin areas are more highly condensed than the euchromatin area. So euchromatin area we can view as a softer area, fleshy area. And these area also, coincidentally, these area contain majority of the genes. This area, the intrinsically permanent heterochromatin area, seldom carry any meaningful genes. That's the difference. But now that we are going to take a look at is the dynamic nature of this even euchromatin area. This area can also change. They don't stay the same, but depending on the circumstances, they change what? Their condensation status. So in other words, even, you are kind of a euchromatin area, but sometimes you can become more heterochromatin. And some other times, if you feel good about it, then now you return to euchromatin, kind of. So that's what I call as a dynamic nature. So generally, we can state such behavior as a given region on a chromosome can reversibly change, interchangeably switch back and forth between euchromatin and heterochromatin. Yeah. Although it applies to this area, the permanent area of this heterochromatin, they don't do that. That's why I call them as a permanent intrinsically. But speaking of this euchromatin area, they can have their relative status. They can still become more heterochromatin, or they can become their original identity, which is euchromatin kind of stuff. And that's very important in what? In their gene expression. Because this tightness and relaxed thing is tightly correlated whether you can function as a gene or not. If you have a highly condensed area, then there's no way you can function as a gene. You cannot express, you cannot make RNA under such physically limited, refined status. In order to act as a gene to produce many RNA, you have to relax first. You have to loosen up, like just a human. You cannot function if you are so tight nervous. Yeah. In order to do something right, first you have to relax and take it easy. So no, forget it. No. So actually, this epigenetics I mentioned several times earlier, probably prematurely. But still, I had to do that because the epigenetics thing, the concept of epigenetics became so important and so like publicized in these days. So what's epigenetics? And definition of epigenetics is change in trait without the DNA sequence change is epigenetics. That's the original definition of epigenetics. Definition of epigenetics, I will repeat. The change of a trait. According to the classical view of genetics, if you want to have any different trait, first, your gene has to be changed, right? How can you change your gene? There is only one way, we thought. Your DNA sequence change. So mutation, yeah. So without mutation, you cannot have any variation in the trait, different traits. But then this guy came up very recently. Hey, it is also possible without any DNA sequence change, you can have any different traits. So that's the concept of this epigenetics. So if you compare DNA sequence, you do not get to see any difference, but still you get to observe actual noticeable differences in your appearance or in any other traits. Then how can you explain this? DNA sequences are the same, but you are different. One such a particular example is actually racial difference. So probably racial difference, although it is not officially declared, but there is no other way but to simply explain using this epigenetics. The racial differences are simply the one such a good example of epigenetic difference. Because if you compare DNA sequence among different races, you do not actually have any corresponding the noticeable DNA sequence change. But everybody can recognize the actual distinctive differences among different races. So this is the definition. Changes in trait without DNA sequence alteration in the gene is epigenetics. How? Yeah, if you think about it, you can still have a different trait by simply adjusting the amount of a gene expressed. So it is up to the gene expression difference. If you have some more gene expression in some occasions, rather than versus a smaller, less gene expression, then it can make like noticeable outside differences. You can get many such examples. So if you compare the gene themselves, but still you can notice. For example, like hemoglobin, once again hemoglobin, as if there is only one gene but hemoglobin. I always take the example of hemoglobin. I'm a hemo guy. I don't know. But anyway, hemoglobin. If you compare the hemoglobin DNA sequences, then you get to find no differences at all. Everybody has the same, exactly same identical hemoglobin DNA sequences. But if some people produce more hemoglobin RNA gene expression, then as a consequence, you will be able to produce more hemoglobin protein versus those who produce only very small amount of hemoglobin RNA. And as a consequence, you get to produce very small amount of hemoglobin protein, then now in terms of its ability to carry the oxygen transport, it's a huge difference. And those who produce more hemoglobin protein will be able to enjoy this more ample supply of oxygen, and then will become active. And those who have a very little amount of hemoglobin protein will suffer from such. And you can think of many such similar examples in terms of this. So still, if you compare this hemoglobin DNA sequence, you cannot point out the reason. What's the difference? They are the same. But you missed something, the amount of the gene expression. That's why this, how much you actually have in terms of the RNA production as well as consequence as a resultant, the protein production will make, a lot of times, will make huge noticeable differences. Okay, I will stop here, take a 25 minutes break, and then we'll continue.